Hey, you know what time it is? It's time to talk more about photosynthesis and specifically what's going on inside the thylakoid. And you know why? Because there's no one else here and we might as well just get this done. So this, if any, will probably be the screencast that most of you will want to watch more than once. There's a lot of detail here, there's a lot going on, and I think once you visit this stuff enough and you take a look at your diagrams, it should make sense to you, but I want to make sure you see how all the pieces fit together. Okay? On this first slide, really important, I can't stress this enough, you have to know that formula, know the whole thing, know the numbers in front, you have to be able to balance it, make sure you know the words, make sure you know the symbols, know the whole thing. Okay? The only thing that you'll see on here occasionally that changes is that right where this says sunlight, sometimes instead of putting sunlight on the reactant side instead of the product side, they'll put it right over top of this arrow occasionally, but it still means in the presence of sunlight, and usually we'll put nutrients in there too because there are other nutrients that are really necessary for this. But for the most part, make sure you are familiar with that formula because that is going to be what I'm going to ask you to produce on your test. Okay? So on this slide, what you're going to see is basically us trying to zoom in on the portion of the leaf or the part of the cell that's most important during photosynthesis. So starting off in the upper left, you're going to see here are some leaves, and we've taken a small box and we've kind of going to zoom in on that box and see part of the leaf, and you can see here's the leaf with all the mesophyll cells, and these are going to be the cells that actually perform photosynthesis for the plant. Underneath that, you're going to see the under part of the leaf, and that's where these stomata are, which contain those two guard cells we had talked about that when they open up can release oxygen and let carbon dioxide in. Also, water can get out of those occasionally. Okay. We're going to take that, and you can see here's the box right here. We're going to zoom in on a mesophyll cell. So here's one mesophyll cell. And what should strike you about that is that it's very green, and that's because there's lots of chloroplasts in there. So then again, we're going to zoom in on one chloroplast that's here and take a look at that, and this is an artist rendering of that, but here's my chloroplast. So inside the chloroplast, the fluid that's in here is called the stroma. I've also got these little hockey puck guys that are stacked up. A whole stack of them is called the grana. Each individual one of these guys is called a thylakoid, and the membrane that encloses them we call the thylakoid membrane. So that's an artist drawing of that. This is actually the real deal down here, and this is a micrograph of that, and you can see they're quite small. This is, this symbol here means micrometer, which is 10 to the minus 6th of a meter. So we're talking about things that are quite small. All of these darkened areas here are going to be the grana, which are going to be the stacks of thylakoids. And really, for most of what we do during the first half of photosynthesis, we're going to focus on one of those thylakoids just to find out what's going on the membrane of that thing. That's where most of the action is taking place. Okay. Okay, I like this graphic a lot because even though it's quite simple, it tells us quite a bit. And as we learn more about photosynthesis, you'll be able to point this out to me. But it basically is telling us if these are the reactants up here and these are the products, it's kind of showing me where each of the reactants is going to end up in the products. And probably the thing that I'd like you to see that's the most important is that when we produce oxygen down here, this oxygen is not produced from carbon dioxide. Probably the biggest misconception that I hear from people learning about this is they think that when a plant takes in carbon dioxide, it's just going to chop off that carbon and it's going to release the oxygen. That's just not what happens. Turns out all the oxygen that is released into the environment actually comes from the water that's put into the plant. So when you go water your plants at home, that's actually what's going to help produce oxygen inside your house. It's not the carbon dioxide in the air. So you breathing on your plant certainly going to help this process along, but all the oxygen that comes out actually stems from the water that's added to the plant. So that's one thing I wanted to point out. These other ones we'll come back to a bit later to learn more about that. Stages of photosynthesis. There are 2 of them, and you will get to love them both, I'm sure, because it's super fun. Part 1 we usually call the light reactions, and that's because those reactions generally take place during the light part of the day. We need sunlight for those things to happen. And so during those reactions, light is necessary. You'll see that we need water during that part of the reaction. Oxygen is released during that part of the reaction. And we're producing 2 other guys, one of which you've heard. You've heard of ATP. You probably have not heard of this guy before. We'll talk about him a little bit later, but he's a type of what we call an electron carrier. Okay, so that's all happening during the light reactions, which that's all going to be what we talk about next. The second half of what happens, which is actually when we make our sugars using this stuff, is going to be something called the Calvin cycle. And I put over here the dark reactions in quotation marks. We don't call it the dark reactions because it doesn't just happen in the dark. It's happening during daylight hours as well, but the process is not totally dependent on darkness or lightness. It just happens all the time. So the Calvin cycle, we call the Calvin cycle. The full name of it is the Calvin-Benson cycle. I guess one of the guys wasn't important enough to keep in there all the time, but essentially those are the two we have to talk about. So ultimately, the first half of photosynthesis, you'll see this diagram down here, is going to look a lot like this stuff, which looks quite confusing, but we'll figure it out. The second half is going to look a lot like this, which again, that one also looks quite confusing, but we're going to simplify that one quite a bit. And the idea is when we're all done is to make sense of both of these things and figure out how they're related to each other and how sugar gets produced, because that ultimately is what all of us animals rely on, is the sugar that's produced during this process. Without it, none of us would be here. All producers pave the way for everybody else, okay? In super duper simplified form, and I do mean super duper simplified, here is what's going to happen for the whole process, okay? So we'll break it down. Here's the general idea of what's going to happen, okay? The light reactions are all going to take place in the thylakoid, around the thylakoid membrane. And basically what happens is light energy is going to make its way into the cell. It's going to go through the membranes. It's going to go all the way through into the chloroplast, and it's going to strike some of the chemicals, some of the pigments we had talked about on the thylakoid membrane. When water gets into the thylakoid, we're going to start the reaction. We're going to start photosynthesis. And as soon as that happens, one of the first things that will happen is that water will be split. We're going to give off oxygen, and it turns out through some of the processes that we'll learn about very soon, two things are immediately going to be produced. And one of those things is ATP, which is energy, and the other thing is this thing called NADPH, which is an electron carrier. And at this point, we've made no sugar. Okay, so we've consumed water, which was part of our reactant. We've consumed sunlight, that was part of our reactant. We've produced oxygen, that was one of our products. But we have had nothing yet to do with sugar, and we haven't done anything in terms of carbon dioxide yet, and that's all going to be coming during the second part. So all this stuff that's happened on the left-hand side is essentially going to be the light reactions, okay? And then during the dark reactions, or the Calvin cycle, this is where we're going to take carbon dioxide that's up here. It's going to go through this cycle called the Calvin cycle, and while we do that, we're going to create sugars, which is ultimately the end product of this whole thing. And then during this process, certain things are recycled, and they move back and forth between these two places. Biggest thing I want you to notice is that the light reactions are all going to take place over here inside the thylakoid. All of the Calvin cycle, or all the dark reactions, are going to take place just within the stroma itself. No specific location, and they're just in the stroma, which is inside the chloroplast. Regarding two of the products of the light reactions, the first half of photosynthesis, we have ATP and NADPH. So ATP is an energy source, NADPH is what we call an electron carrier, and you'll see why that's important a little bit later. Okay. Well, it turns out the structures of these things are incredibly important to their function. So let's focus on ATP first. All right. ATP, adenosine triphosphate. Adenosine is the base that's found in that. The T stands for tri, which means 3, and the P stands for phosphate. So when you look down on the bottom left here, we've got a sugar, that's ribose, and we learned about that earlier. We do have this structure right here, which happens to be a base, which is called adenosine. It's got 2 rings on it. And then off to the left, you can see I've got 1, 2, 3 phosphate groups. And the key part of this is that ATP, because it has these 3 phosphate groups that are all negatively charged, each one of these phosphates does not want to be near another phosphate. They're all the same charge. They're all repelling each other. If you can imagine being chained to 2 other people that you absolutely hate for an entire day, you know how this feels, okay? These phosphates want to get away from each other in the worst way, but they're being held together by these bonds. And so if we can release one of those bonds, if I could take off if you were handcuffed to somebody, if I could take off one of the cuffs, you'd be much happier because you'd only have 1 person around you didn't like instead of 2, and you'd be a lot freer to do things. So it turns out in ATP, the one bond that's always going to be broken, and it's going to turn it into ADP, adenosine diphosphate, and we usually put this inorganic phosphate that's floating around, is that if you can imagine a magic pair of scissors is going to cut this bond right in here, okay? And when that happens, this phosphate becomes free, and this is going to be ADP. That will be regenerated through some reactions that we'll see, but that's essentially whenever this bond is broken right here, this is between the 2nd and 3rd phosphate sticking out, all kinds of energy is going to be released to do stuff, make stuff. And so that's where all the energy is coming from. On the other side over here, you're going to see this neat looking molecule called NADP plus or NADPH plus, H plus, and the H plus is a hydrogen ion. That's got a specific name which we'll get into later, but you can see the structural difference between this guy and this guy. One of them has this extra H hanging off of it, and you can see that right here and right over here. As far as this other piece that's over here, we'll get into that a little bit later. You can see that happens to be something that was ripped off the top here and kind of moved a little bit. More importantly, what I want you to understand is that this guy right here, it does something, it actually carries electrons, and so we'll talk more about why that's important as we get through the reactions a bit later. All right, so another very general diagram to lead us into the next idea, which is that inside the chloroplast, this light that's up here, white light from the sun, is going to travel down into the chloroplast. As soon as it gets into the thylakoids have these chemicals that are called chlorophyll. They reflect green light. You can see that taken off into the distance. All the other colors get absorbed in there, and they're there to do stuff, and that stuff is what we're going to talk about next. So in terms of the absorption of sunlight, and this is a little bit of chemistry lesson here, but whenever sunlight gets absorbed by some particular pigment, really what's happening on the atomic level is that an electron, which normally is circling the nucleus at one particular energy level, when it absorbs energy from sunlight, here's some sunlight coming down and it strikes this electron, electrons tend to get excited by gaining energy, and when they gain energy from a photon, a little chunk of sunlight, they will jump up an extra energy level, and it takes more energy to circle at this higher radius out. So any atom that has an electron that's at an excited state means that the electron is actually circling at a higher level than normal, if you want to think about it that way. Which on the flip side of that means that if this electron jumps down a level, it's actually going to emit light or give that light back off. And in fact, that's how we have light bulbs that actually work, is because we excite electrons, they release them, and that's the light that we get from those things. So this diagram, which looks quite complicated, is talking about that same idea, and it's saying that whenever you have a chlorophyll molecule, which is right here, okay, that chlorophyll molecule is made of atoms. When sunlight strikes the atoms within that chlorophyll, some of the electrons get excited. They jump up a level. Now on this one, we don't have the circles that are there, but you can see the diagram showing that it's jumping up a level. That's an excited state. Now if that electron were to jump back down to where it came from, when it jumps back down, just like if I were to jump off a table and onto your head, I would be releasing energy. Well, the energy that's being released here is going to be, number one, heat, and number two is going to be light, which is a photon. The distance that the electron falls is going to correlate with a wavelength of light. Okay? So different wavelengths of light that we see are actually produced by electrons jumping different levels within different types of atoms, which is kind of a neat thing. Okay? So let's move on to our next slide. All right, big time. This is where it all begins right here, really. So we've got the photosystems, and those are going to be the two chemical regions where we're going to find our chlorophyll, or whatever photosynthetic pigment we're talking about. In this case, we're going to talk about chlorophyll. Okay. So we've got two of them, and I always say them silly. I say photosystem with my teeth together just to help myself remember them, and there's two of them. I know that's dumb. The first one that we're going to talk about is called photosystem 2, and there's another one that I call photosystem 1. And they're similar. They absorb slightly different wavelengths of light, but let's walk kind of through what goes on with this thing, because there's actually quite a bit going on for the light reactions here. So at the bottom of the page here, you're going to see photosystem 2, and then over here photosystem 1, along with a bunch of other things in between, and we're going to simplify those and give them just one name, really, or a couple different names, maybe two that you'll have to know, but other than that, they're general things called electron carriers. So let's move on and find out what's going on here. Okay. First thing, it's not on this slide, and you'll see that I've used my Spanish here because I'm very fancy. But whenever we start this process, one of the first things that has to happen is that water inside the cell needs to get inside the thylakoid. Okay, so it's going into the chloroplast, into the thylakoid. Once it gets into the thylakoid, it's going to undergo something called photolysis. And if you break that apart, photolysis literally means light cutting. Okay, so what's going to happen is during photolysis, we're going to take the particles of water and break it apart into everything we possibly can, just about. Okay, so when I take water, which is right here, and you can see the blue is our oxygen and the white is going to be our hydrogen atoms. The first thing we're going to do is kind of cut these bonds right here, and that means I'm going to get my oxygen right here, and you can see it says half O2. Oxygen is a diatomic gas. It always hangs out in pairs together, so this is half of that. That's an oxygen atom. Here's my hydrogen atom, okay, and what really happens is hydrogen happens to be made of 1 proton and 1 electron. That's it. So it turns out that hydrogen atom actually splits itself into each one of those entities. So you have oxygen here, you have a hydrogen nucleus, the hydrogen ion, and then the electron that used to go around that, and they're now separated. Since water has 2 hydrogen atoms, we get 2 of these things. So when we're all done, all this stuff down here, we get an oxygen atom, we get 2 hydrogen ions, which that should be a plus up there, and then we're going to get 2 electrons to go along with that. And these electrons are really important to get things rolling, because without those things, none of this would really happen. We need those electrons. It's almost like the fuel that runs the light reactions. Okay. So this is going to look a little spooky here, but what we're going to start with is down here. This is where photolysis is going to take place, and I'm putting a P there for photolysis. So our water's made it inside the thylakoid down here. That's where photolysis is going to take place. So once that's all taken place, what we need is sunlight. Sunlight's going to shine, and when it comes down, a beam of sunlight is going to hit photosystem 2. And in doing that, what we need to do is make sure that there are electrons present here to get excited so they can go up to their next level. But if I don't have extra electrons around, that can't happen, okay? It's like playing whack-a-mole without the mole. If you don't have a mole, you can't hit anything. You're smashing the table. Well, if I have no electrons, nothing's going to happen here. Sunlight's going to hit this thing. There's going to be no reaction, okay? So the key is once we get all of the necessary things in place, we're going to be using both our photosystems, which are 2 areas where there's chlorophyll present. There are some electron carriers, some transporters in between there. And ultimately what we're going to end up doing is trying to create a concentration gradient with hydrogen ions, meaning I want to get more hydrogen ions on one side of the thylakoid membrane than the other. And by doing that, we'll find out that if we can cram all these hydrogen ions into one place, they're going to want to go across this membrane, but they can't do that unless they go through a specific protein channel, right? And this is going to be an ion transport. Well, it turns out there's an enzyme that is hooked up to one of these protein transport systems that when these hydrogen ions come flying back through to trying to equalize their concentration, when they do that, it's like me going down a slide. If you were at the bottom of the slide and you didn't get off the slide and I came down, I can guarantee you it would hurt. I weigh a lot. And I came down, and if I kicked you, it would hurt. I'd be releasing energy. Well, as these hydrogen ions come through these proteins, they release energy, and the energy that's being released ends up taking adenosine diphosphate, takes the extra phosphate and tacks it back on there, even though it doesn't want to go there because they're all the same charge. And that's going to end up making ATP, adenosine triphosphate, which we're going to use later on for another part of the cycle. So this is really where it's at. I lied last time. And this is what's called the famous Z-scheme of photosynthesis, at least what I call the Z-scheme, because when we're all done, it's sort of going to look like a Z if you use your imagination. So we've talked about all the necessary players here. The big purple thing is going to be photosystem 2. Within photosystem 2, there are going to be chlorophyll molecules that are there. The chlorophyll are going to be the molecules that absorb the sunlight, so they're going to need to have electrons. So way down here, when you see this light striking one of these little areas here that are green, what has to happen is we have to make sure that all of these things have extra electrons. Now, if I keep exciting electrons and making them go up, we're going to find out that they're actually going to travel on this diagram. They don't actually do this the way that it's shown here, but they're going to go up and they're going to actually travel away from this area. And if I was sitting, or I guess this way, if I was playing tennis and I had a machine that was shooting tennis balls at me, eventually those tennis balls are going to run out, and I'm going to need to pick up all the balls and put them back in the bucket, so if I want to keep practicing, they'll be there. Well, if I want photosynthesis to keep taking place, I got to have more electrons supplied to this whole photosystem, and that's where the water comes in. All the water, which is going to be down here, okay, and I can't write very well with this thing, but this is going to be H2O down here. When that gets split into oxygen, hydrogen ions, and electrons, those electrons are going to end up going bloop, right onto photosystem 2. And that's where we're going to have our electrons going from. So it's shown a little bit differently here than I like to think about it, but water is split. It creates the electrons that are here so that when we have these light photons that shoot in and they excite the electrons, they jump up, and you're going to see this jump up to the top of this molecule. Again, that's not exactly what happens, but for a diagram, that's a good way to think about it. As soon as that happens and it gets to the top of the photosystem, the next thing is that that excited electron is going to be passed off to another molecule. This PQ that's here is something called the plastoquinone, and the other one, I'll be honest with you, I can't remember the PC one. But this whole thing we're going to call the electron transport chain. The purpose of that chain is to take this electron that was excited from here, and because that electron had a lot of energy, it's going to be moved someplace else. And by moving it, you'll see that it's moving down a little bit. And I want you to think about that as moving down a slide, because as this electron moves down this slide, it's slowly going to release energy. Okay? The important part of this is that while that's happening, there's a whole other thing going on where as this energy is being lost, we're using energy here to do some active transport of another ion. And I'm going to wait until we're all done to show you where that takes place, but that's a really important part of the process. So at this point, we've seen light come in, strike chloroplast, excite electrons, the electrons that came from water originally, okay? That electron is going to move down this electron transport chain. It's slowly losing energy, which is being used for something else, so there's no waste of energy. Once that moves all the way down the slide, you can see it's going to reach another photosystem, and we're going to call this one photosystem 1. And it turns out this one, P700, tends to absorb wavelengths of light around 700 nanometers, whereas this one over here is going to absorb sunlight around 680 nanometers. That's the difference between those two. They absorb slightly different wavelengths of light. As that electron slides down this slide, the way I like to think about it is that when it comes all the way down, it kind of knocks another electron loose, which gets it excited. Light's also down here exciting other electrons. One electron is going to get very excited again, and because it has lots of energy, that means we can use it to do things. So we have an excited electron that's up here. After that electron is boosted up to the top of this thing, it's going to be passed off to this thing called NADP+. At this point, I know that's a bit of mystery for you guys, but essentially what this is an electron storage facility. And it's going to take it from here, which is on the thylakoid membrane, and take it to a completely different area called stroma, which is where we're going to have the second part of photosynthesis take place. So essentially, you can sort of see this sort of Z scheme that's here, all right? The important thing that I left out has to do with production of ATP. So let's talk about that real quick. You'll see this a little bit better in one of the last pictures I'm going to show you today. But it turns out whenever this electron is sliding down this slide and releasing energy, the energy that's being lost is going to be used to pump hydrogen ions from outside the thylakoid and pump them into the middle of the thylakoid. Now the reason why it has to pump them, if you recall, when water was split over here, and I'm going to say down here where I'm coloring in, one of the things given off was oxygen. That's a waste product. One of the things that was produced from that photolysis was the electrons that are going through this cycle, and the last thing happens to be these hydrogen ions, which we generally would write something like this, just an H with a little plus next to it. Okay? Well, the more water we split, the higher the concentration of this hydrogen ions that are there. So I only put a little up arrow right here. I know it's really messy. But essentially that means that since there's already a high concentration of hydrogen ions in here, when I have electrons sliding down this chain, the lost energy from that is going to pump even more of those things inside. And I'll show you in a minute why the thylakoid wants that to happen. But what we really want to do is produce as much of this hydrogen ion concentration as we can inside the thylakoid. That's going to be really important for making ATP. So, quick review of what's happening here. Let's kind of move through this here. This is fairly simplified, and it's showing you what's happening with energy. So I start off with a photon, which is going to sort of elevate this electron, get it excited. The electron is going to move down the slide, and in doing so, you can see this little mill. It's just showing you that some of that lost energy is being used to do stuff. Okay, don't be confused about the direction of this ATP that's being produced. We'll talk about that in a minute. But really what's happening is hydrogen ions are being pumped into the thylakoid right here. Once the electron makes it down to here, turns out another photon is going to jack that thing way up, get it excited, which really means it's moving up another level. And that excited electron is going to become part of this thing called NADPH. which is an electron carrier, which we're kind of going to get to a little bit later. So the diagram that I did in class is going to look a lot like this one, and this is probably the only one that you saw if you were in class and haven't watched this yet. In this diagram, this has everything you need to know about the light reactions, okay? The key is I have water that happens to be right here. Through the process of photolysis, it gets split into oxygen, hydrogen ions, and electrons. The electrons supply this whole electron transport chain so that when light comes in and strikes the photosystem, it excites an electron. That electron has more energy than it used to. It gets passed off to this series of electron carriers called the electron transport chain. It's not drawn this way here, but you want to think about this gradually moving its way down over to here so that it's gradually losing energy. As it loses energy from outside the thylakoid, we're going to pump more H pluses into the center of the thylakoid, thereby making the concentration of hydrogen ions inside the thylakoid very high. So we'd want to have this up arrow right next to this to say there's lots of hydrogen ions there. Once this last electron makes it to this first photosystem 1, virtually the same process occurs. The electron that's here, light hits the photosystem, it excites it, it moves up, and that second electron is going to move off and eventually become something that becomes part of this molecule right here, which at this point we've just said that that is something that is going to be used as an electron carrier. Okay. So at this point, way back in the beginning of the slide, we said two of the products of the light reaction. One of them is going to be NADP plus and 2H plus, or excuse me, this stuff here, NADPH plus H plus. That's an electron carrier. The other thing that's being produced is ATP, and that whole process has to happen through this protein right down here. So since there's a high concentration of hydrogen inside this thylakoid membrane, they really want to get out, and the only place they can get out, because they can't get out through the phospholipid bilayer, is to go through one of these ion channels. So this particular ion channel right here happens to be hooked up to an enzyme called ATP synthase. It's so named because it creates ATP, but as those hydrogen ions shoot out through this protein, they release energy, just like me sliding down the slide and kick you in the back. And so when they slide down this slide and they leave the whole thylakoid area, they release energy. The energy that's released is used to take an ADP molecule and a phosphate that's hanging around, and it takes this phosphate and kind of glues it right back on there and bonds it. It makes ATP. So by the end of the light reactions, we've created NADPH, an electron carrier, and ATP, energy needed to do stuff, which as you can see from this little slide up here, we're going to need that ATP to create sugars during the Calvin cycle. And in just under the shade of a half an hour, that is the basics of the light reactions. Obviously, this could go a lot more quickly if I was sitting next to you explaining this to you, but to be as complete as I can and thorough as I could about it, that's what we've got. So the next section that we'll talk about is going to be the Calvin cycle. So come back to check out what's important about that stuff. I'll see you then.